Hello students, now we will learn about monocot endospermic seed. Here we will take example like maize, rice, etc. We'll draw the structure, we'll see the labelings. This is the endosperm, stored food. Endosperm. Here, in case of monocots, the endosperm is made up of starch, means stored food material is starch. Next. This is epithelial layer. This outermost part is pericarp. Pericarp in case of monocots. Now, seed pod in case of dicots we have seen it is divided into testa outer layer and tegmen inner layer. But here the seed pod is fused with that of pericarp. Pericarp is a combination or composed of, what are its parts in pericarp? Exocarp, mesocarp, endocarp. So these three layers are constituting the pericarp. The innermost layer is aleurone layer. Aleurone layer is a proteinaceous layer. Here will be the cotyledon of monocot called as scutellum. And here will be the embryonal axis. In embryonal axis, the difference between dicot and monocot is, in case of dicot, we are labeling the edge part as one is plumule, one is radical. But here, the tip part is different. It is, if it is giving a rise to the shoot part, then it is coleoptile. And here this is coleorhiza. And this part is plumule, which develops into shoot part. And this is radical. So these are the labelings in case of monocot endospermic seed. Even if we take the rice grain also, still aleurone layer can be the second one. Outermost one, pericarp. This will be the reserve food material, stored endosperm, full of starch. Now, scutellum will be this one. Embryonal axis. Now, at the tip part will be, this is coleorhiza and this is coleoptile. And here this will be radical and this will be plumule. So these are the labelings of monocot endospermic seeds. Next we will see about monocot non-endospermic seed. Example already we have seen. Non-endospermic seed, also called as exalbuminous seed. Orchids. So this is scutellum. This is embryonal axis. The same way the tip parts. One is coleorrheza. One is coleoptile. So here will be plumule. Here will be radicle. So this is the labeling part of orchid's seed. Next we will see one more point, that is perisperm. Basically what happens? Before fertilization of the ovule, the reserve food material in an ovule is nucellus. But later after fertilization, means double fertilization, triple fusion, it will be converted into endosperm. Later the cotyledons and endosperm together will be working as stored food material. Cotyledons will be taking another function like developing into embryonic leaves or primary leaves, and endosperm will be fixed as the barrier between the seed development and germinating seed, and also endosperm is completely the reserve food material for the germinating seed. But sometimes nucellus is not converted into means endosperm. It will be undergoing many number of divisions. It will be in the form of nucellus only, undergoing many number of mitotic divisions. In some seeds like black pepper and beetroot, nucellus is not converted into endosperm. So even after fertilization, the remains of nucellus are called perisperm.